Alrighty, so before we start this video, let's just go ahead and refresh eukaryotic and prokaryotic cells. So the cells at the top, the two very complex ones that have organelles and a nucleus, those are eukaryotic. That's plants, animals. At the bottom, I've got a prokaryotic cell. Remember, that's bacteria. There are four things that all cells have in common. Remember, we said cell membrane, cytoplasm, DNA, and ribosomes. Don't hate runny chocolate chips. If I notice, cell membrane and cytoplasm, their jobs are basically for support. So cell membrane, the plasma membrane, keeps everything together. Cytoplasm is the gel that keeps everything from sloshing around. DNA and ribosomes, though, they're not for support. They're actually there for very important jobs. And today we'll go ahead and talk about what those jobs are. Why do I need DNA? Why do I need ribosomes in all of my cells? Just as a quick reminder, for DNA in pro, it's floating in the cytoplasm. They don't have a nucleus. They don't have organelles. Ribosomes are also just floating in the cytoplasm. Those are the little dots. In U, they're complex. They keep their DNA in a nucleus, and their ribosomes are the little dots that are either free-floating or they are bound to the endoplasmic reticulum. So they are either bound or they're free-floating in the cytoplasm. Alrighty, so let's go ahead and review DNA structure really quickly. So if I look at DNA, remember it is what we call a double helix. That means a double helix is two twisted strands of DNA. So it is two strands twisted together. If I look in this picture right here, here is strand number 1, here is strand number 2. What is holding those two strands together? Hydrogen bonds. The letters in the middle, the A, the T, the C, and the G, that's what's actually being held together. So if I look, I have the letter A, and there is a hydrogen bond between it with the letter T. There is a letter C with hydrogen bonds in between it with the letter G. So A always matches up with T, C always matches up with G. Now notice I wrote 3 lines in the middle of C and G, and I only wrote 2 lines in the middle of A and T. So that means there are 2 hydrogen bonds, you do need to know this, between A and T, and between C and G there are 3 hydrogen bonds. Alrighty, so just going back, we said it's a double helix made of 2 strands of DNA. If I look at the structure of DNA, it is made of a bunch of nucleotides. So what is a nucleotide? That is its monomer. It has a chimney, a house, and a garage. Remember that the chimney is called, let me erase this stuff. The chimney is called, erasing. The chimney is called a phosphate. The house is called a sugar, and the garage is going to be the nitrogen bases. And again, going back, the sugar and the phosphate, that actually makes up the support. That makes up the backbone. So that's the green part. It holds it all together. If I look, though, at the nitrogen bases, that is the important part. Those are my letters in the middle. That's my A's, my T's, my C's, and my G's. And again, just as a refresher of what I just said, A always goes with T, C always goes with G. The easy way to remember that is apple, tree, car, garage. Alrighty, so why do I have to have DNA? Why is it important? The code of DNA actually tells me how to make proteins. And we're going to go ahead and talk about how we read that code in a second. But just remember that DNA is super important because it basically tells me how to make the proteins that decide how my body looks. So for example, if you have dark colored hair, you are producing a protein that actually gives you the color. Well, where did your cells know how to make that protein? They have it in their DNA. Their DNA tells them, make this protein for dark colored hair. So your DNA tells you how to make all the things that your cell needs. Alrighty, sometimes when I'm copying my DNA though, I can make mistakes. So for example, mutagens, which are chemicals, UV light or radiation, can actually mess up my DNA. I can also, again, mess up my DNA during DNA replication. Remember, the word replication means I'm making a copy. If I mess up my DNA, then it will get passed on to my kids if it happens in my gametes. Remember, the word gamete means sex cell. If I mess up my DNA in my somatic cells, my body cells, it does not get passed on to my kids. So for example, if I accidentally dump my hand into a mutagen, I am messing up my somatic cells. That messed up DNA won't get passed on to my kids. But if my whole body gets dunked into a mutagen and a chemical and all of the cells in my body are changed and mutated, then it will get passed on to my kids if my sex cells get damaged also. All right, so let's talk about some of those mutations. What are the mutations I will see? So right here, this is my original DNA. This is what I want to see in my body. Remember, ladies and gentlemen, I read 3 letters at a time. Each 3 letters codes for 1 amino acid. So what I'm circling, that's the name of 1 amino acid. So up here in gold, this is what I originally wanted. Let's go ahead and look at the first one. I wanted TAT, that's what I got. So the first one is fine. The second one, I got CAT, that's what I wanted, I'm okay. The next one is CGT. If I look up at the original, I did not want CGT, I wanted CCT. So there was a mistake. What I ended up doing was I swapped 1 letter for another. So I substituted 1 letter for another. A substitution, this is a type of mutation, it is called a point mutation. What happens in a point mutation? I only changed 1 amino acid here. Because if I look at the other 2, LYS, LYS, VAL, VAL, I got the same second amino acid, so I'm fine. I just changed this 1 amino acid. If I change 1 amino acid, then what will end up happening is that my protein could have a different structure, meaning it could work differently. But notice, this isn't that bad because I only changed 1 amino acid because I only swapped 1 letter for the other. In the next example, we're going to change my color. My next example, TAT, I wanted TAT, we're fine. The next one, CAT, CAT, we're good. Now I have CGC. That C, something happened to it. A letter got squished in between. That letter that got squished in between, the letter G, that is called an insertion. So I pushed that letter G in there when it didn't belong. Now remember, I read 3 letters at a time. So instead of reading CCT, I'm going to read CGC. If I read CGC, I get arginine. And now, remember, I need to read the next 3 letters, so I read TAA, which gives me a stop codon. That literally means stop making the amino acid. So what happened to the letters? Let me do this in blue. What happened to these letters? Do I read them? No. Do I make the rest of my protein? No. If I don't make the rest of my protein, will it work the way it's supposed to? No. Could this harm my body? Yes. So insertions, if I push a letter into DNA that didn't belong, this is called frame shift. And the reason why we call it frame shift is because I shift my letters when I read them. So now notice how I had to move everything over, and because I moved everything over, I shifted. It messes up all of my amino acids. This is really bad. Alrighty, the last example, TAT, let me change my color. TAT, this is what I got, this is what I wanted, I'm fine. The next one, C blank T, I deleted a letter, and if I delete a letter, again, remember, I changed the way I read it, so now I read CTC. That gives me the amino acid leucine. I didn't want leucine, I wanted HIS, so 1, my amino acid is messed up. If I look at the next one, let's do this in purple. CTA. Again, I had to shift everything over and I got leucine. I didn't want leucine. It's messing up all of my amino acids. If I look at the next one, I have to read AGG. Again, I'm shifting everything over. I'm messing up all of my amino acids. This will change all of that protein, so that protein won't work the right way. This is, again, frame shift. So there are 2 types of frame shift. There's an insertion and a deletion. You need to be able to tell the difference between the 2. Obviously, deletion, I delete a letter. Insertion, I insert a letter. But remember, frame shift is way worse than point because I'm moving everything over, so I'm changing that whole protein. Substitution, I'm just changing 1 amino acid in that protein. Alrighty, how do I organize DNA? I organize DNA in the form of chromosomes. So what is a chromosome? A chromosome is just folded up DNA. We section off DNA into genes. What is a gene? Make sure you have this written down. A gene is an area of DNA that codes for 1 protein. So in this picture, I have 2 genes. So that would mean I'm coding for 2 different proteins. For example, let's say gene 1 is for your eye color. Let's say gene 2 is for your hair color. So they tell me how to make different proteins, but they're on the same chromosome. If I look at this picture, I've got these colored bands. Each one of those represents a different gene, and we usually give them names so we can tell the difference. So for example, this one might be called E, this one might be called H, because I'm talking about different genes. Alrighty, sometimes my chromosomes can have mutations. This is the original DNA that I wanted, the original chromosome. I wanted it to be in this order, A, B, C, D, E, F. In a deletion of my chromosome, obviously I deleted the letter B. Now, am I actually just deleting one letter? No. Here in this picture, I'm actually deleting a whole chunk of DNA. I'm deleting a whole gene, meaning my body won't know how to make that protein at all because the gene got deleted for it. This is so much worse than what we talked about on the last page, because instead of just changing one amino acid or just changing one protein, I'm permanently deleting it, meaning I will not know how to make that. My body might have some type of problem. The next one is, here's the original I wanted. I duplicated. A duplication means I copied. So I got B twice. I copied that DNA. So now I'll make extra of that protein every single time. If I make extra of that protein, that can cause a problem in my body. The next one is inversion. The word inversion means to flip. So notice how my chromosome got all twisted up. My genes are going to get all twisted up and messed up. So I got A, E, D. I didn't want that. I wanted A, B, C. And now I have C, B, F. So all of my order is messed up. This makes it really hard for me to find what gene I'm looking for. All right, the last one is called translocation. Translocation is when my chromosomes that are 2 different chromosomes, they are not homologous. Remember, homologous means a mom and dad copy. This case, non-homologous, they get mixed up and my letters get swapped. If I swap letters, now again, it makes it really difficult for me to find what I'm looking for, and I might not be able to find it and make that protein. Again, if I don't make that protein, my body won't work the way it's supposed to. Alrighty, just as a refresher, when you were created, a sperm and egg came together. Each one of those is called a haploid cell, and it formed a zygote. A zygote is a diploid cell. What does the word haploid mean? What does the word diploid? Haploid means you have half the DNA, meaning that you only have 1 copy of every chromosome. If you are diploid, di means 2. So you have a mom and a dad copy of every chromosome. So what I just circled for you, that is what diploid looks like. A mom and dad copy of every single chromosome. If I were to just circle, let's do this in blue. If I were to just circle this chromosome right here that I'm circling in blue, that is the dad copy of the chromosome. That would have been haploid. So in blue, that's haploid. In green, this is diploid. Alrighty, really quickly as a refresher for you, in the last video we talked about mitosis and meiosis. So for mitosis, remember, I'm taking all the DNA inside of my cell, I'm copying it, and then I'm going to split into 2 cells that have identical DNA. So both of these cells are going to be diploid, meaning they have 2 copies of every chromosome, a mom and a dad. For meiosis, I am going to take all of my DNA, copy it, I'm going to do crossing over, which gives me variety, and then at the very end of 2 sets of cell division, I'm going to get 4 cells that each have half. So they are haploid. Alrighty, real quick, let's talk about protein synthesis. The word synthesis means to build or to create, and in this case, I'm building or creating protein. Remember, there are 2 steps to this process. The first step is called transcription. The second step is called translation. Transcription, with a C, is building mRNA. So I'm going from DNA to mRNA. And just as a quick refresher, DNA and RNA have some differences. The first difference is that DNA is double helix and it has thymine. RNA, on the other hand, is single-stranded and it has uracil. Alrighty, so for step 1, I'm reading DNA and I'm building mRNA. If I'm reading DNA, this step has to happen where the DNA is. So in bacteria, which is a pro, transcription happens in the cytoplasm. because if I look, my DNA is floating in the cytoplasm. In eukaryotic cells, like y'all, this happens in the nucleus because it happens where the DNA is. For translation, what I am doing is I'm reading that mRNA and I am building protein. I am building chains of amino acids. So for translation, it has to happen where the ribosomes are. The ribosomes are in the cytoplasm for bacteria. Phone alarm, sorry. It has to happen in the cytoplasm of pro. For eukaryotic, it has to happen in the cytoplasm as well. So transcription happens where the DNA is, so that's cytoplasm for pro, that's nucleus for you. For translation, it happens where the ribosomes are, that's cytoplasm for both. All right, let's actually look at these processes. So remember, ladies and gentlemen, this is DNA to mRNA, so I know I'm talking about transcription. The way that I do this is my DNA sequence, it will be the complement to it. So if I have a T, complement to T is always A. Complement to A, it should have been T, but RNA doesn't have T, it has U instead. Complement to C is going to be G, G, U, G, U, C, U. Alrighty, again, in this picture, I have a nucleus, so this is going to be a eukaryotic cell. If I were talking about pro, this would just be happening in the cytoplasm. So I'm going from DNA to mRNA. Alrighty, the next step is called translation. Translation is happening where the ribosomes are. Ribosomes are going to read mRNA and they're going to build amino acid chains. Remember, amino acid chains? That's called a polypeptide or a protein. Either word is the same. Alrighty, how do I do this? How does the ribosome do this? Basically, I have to use my codon chart. What is a codon chart? Remember, I read 3 letters of DNA, I'm sorry, 3 letters of mRNA at a time. So my first letter is A. So I look right here where it says first letter. A is right here. So all of these in the box have A as their first letter. Everything in the box is the name of one amino acid. Alrighty, my second letter is U. So I look up here at the top where it says second letter, U, I follow it down. Look where green and yellow mix. That's where I'm trying to pay attention to right now. My last one is G, so I follow where it says third letter, I follow G across. Where do all 3 of my colors meet? Right there where it says met. So my first amino acid is met. Alrighty, let's go ahead and do the next one. So for my next one, again, we said that my first letter is G. So I go down here where it says first base, my first letter is G. My next letter is going to be U. So I go up here where it says second letter, follow U down. Look where green and yellow meet. And then my last letter is G again. So where does it say third letter? I go down here to where G is. I follow it across. Where do all 3 of my letters meet? That would be V-A-L. Alrighty, let's go ahead and do our last one. My first letter is going to be A. So again, I follow A across. My next letter is C. So I go up here to second base. I follow C down. Where do my 2 colors meet? And then my last color is, or sorry, my last letter is going to be U. So I go over here to third letter. I follow U across. Where do all of them meet? At L-L-E. Yeah, we're good. Alrighty, you gotta double check yourself. Alrighty, so for this protein that I'm making, these are the names of the amino acids that would create it. Alrighty, do I need to make all the proteins I know how to make all the time? No. If I made all the proteins I knew how to make all the time, I would waste stuff. Imagine you have a recipe book, and if I only want to cook one thing, I only make that one thing. But if I have a recipe book, and just because I have it, I have to make every single dish all the time, that would waste stuff. So that's not the way we do things. If I have all the DNA to make all the proteins, I don't make them all the time. I only make them when I need it. So let's say, for example, you can break down milk. If you drink milk, then your body needs the enzyme, the protein, that can actually break it down. So in your body, if you drink milk, let's say that's what this little plus signs will be, then what I do is I will take this thing called a repressor off of my gene. So this line right here, that's my gene. If I drink milk, then the repressor will fall off. If the repressor falls off, then that means RNA polymerase can read the gene and build an amino acid. So remember, oh, by the way, for transcription, go back up. RNA polymerase, I forgot to mention this on that slide. RNA polymerase is the name of the enzyme that actually makes RNA. So it's the one that reads DNA and makes RNA. So if I drink milk, the repressor falls off of this gene. My RNA can actually read the gene and it will build the mRNA I need to actually make the amino acid to break down the milk. What is a repressor? Why does it need to fall off? If I repress something, that means I'm holding it down. So the repressor stops RNA polymerase. So let's say, for example, you didn't drink milk that day. Do you need the enzyme that breaks down milk if you didn't drink milk? No. So my repressor stays on the gene. RNA polymerase cannot move. If it cannot move, I don't make the mRNA. If I don't make the mRNA, I don't make that protein. So if I didn't drink milk that day, I don't need the protein to break it down. If I do drink milk, I do need that protein, and so the repressor falls off. The RNA polymerase can actually read this gene and it will make the mRNA so I can make that protein. So again, the idea is, do I make all of my proteins all the time? No. I only make the ones that I need. So on the same note, if you are made out of a single cell, that's how y'all started. Remember, sperm plus egg made a single cell. You now have millions of cells in your body. Where did they come from? Your cells did mitosis. So you started as one cell, it did mitosis, and now you have millions of cells. But if they all came from one cell, then that means they all have identical DNA. Because remember, mitosis, I make identical copies. So if I have identical DNA, why does my skin cell look and work different from my bone cells or my muscle cells or my hair cells? Why do they work and look different? If they have the same DNA, they know how to make the same protein. But remember what I said on the last slide, I don't make all of the protein all of the time. So what I do is something called cell differentiation. Cell differentiation means that I'm differentiating. I'm making something different. So what I do is, let's say, for example, here is the same sequence of DNA in both pictures, but there's a green gene and there is a orange gene. If I want to be a neuron, meaning I want to be a brain cell, I'm only going to read the genes that code for protein that I need as a brain cell. So I'm only going to read, for example, the green gene, and I'm only going to make the green gene proteins. I become a neuron, or I work like a neuron. If I want to be a skin cell, that's what epithelial means. If I want to be a skin cell, do I need to read the same genes that a neuron makes? No, I don't need to read them. I don't need to pay attention to them. So I'm going to ignore the green and I'm only going to pay attention to the orange. I will only make those proteins that orange tells me how to make. Therefore, I become a skin cell. So in your body, even though every single cell in your body has the same DNA, what we're actually looking at though is the fact that we are reading different genes and making different protein. Alrighty, last little example on this. If I look at these bunnies, they are the same type of bunny. Why does one have black ears, feet, and tail while the other one is all white? So if this bunny, the black one, was born and raised in a cold environment, if you go outside and it's cold, the first things that get cold are going to be your nose, your hands, your feet. If you had a tail, it would be your tail too. So what happens is the genes in these areas, the cold areas of the bunny's body, they get turned on to produce the protein that gives it color. So for example, in this bunny that was cold, its feet, its tail, all of those parts of the body, those cells had the gene turn on and it started making the color, therefore the ears and tails and nose became dark. This bunny was raised in a warm environment, nothing was cold. Because it was not cold, the genes for color stayed off. So again, I had a repressor, it blocked it, RNA polymerase did not make the RNA, and I did not read it and make protein. So the bunny stayed all white. So theoretically, and people have tried this is why we know it works, they've taken one of these bunnies, they put an ice pack on its back. So again, we said if it was cold, then it would start to turn on the genes, I would make that protein, and the bunny now has a brown patch on its back because those cells right there where it was cold started turning on and producing that gene. Alrighty, that's it.